In today's video, we're going to be discussing hemoglobin. So, as some of you may or may not already know, hemoglobin is a globular protein which contains heme as a prosthetic group. So, what is a globular protein? Well, it's a water-soluble protein which forms colloids in water, and that means it's able to disperse or spread across a solution microscopically. And it has a tertiary structure, which we've mentioned here, and it has hydrophobic amino acids in the interior of the molecule and hydrophilic amino acids bound outwards. So this means that there's water-hating amino acids in the inside of the globular protein and water-loving amino acids bound outwards on the exterior. So this means that there can be dipole-dipole interactions with other solvents. This is a feature of globular proteins. So this is heme, which I've got here on the right. This is heme B. There's various different types of heme, and they have different structures, but this is heme B, the most common one. So as you can see, there's iron in the center of the molecule. This is Fe2+, and this iron here can form 6 additional bonds. So we have one here, one here, one here, and one here. Those are 4 bonds to the porphyrin nitrogens, and then the iron can form an additional 2 bonds. So one of them will go to a side chain of histidine from the globin itself, so from the globular protein, and this helps to stabilize this heme with the actual globular protein. And then the final position is for the iron to bind oxygen. So the final position is to bind oxygen. So what about hemoglobin itself? Well, it's found in the red blood cells, and its main function is to transport oxygen from the lungs to the capillaries of tissues. And hemoglobin A is the most common in adults. There's various forms of hemoglobin. It's a bit complicated to discuss it in this video, but the most common one is hemoglobin A. And hemoglobin itself has a quaternary structure, so it consists of 2 identical dimers, alpha beta 1 and alpha beta 2. And remember, these are identical as well. So it consists of 2 subunits here and 2 subunits here, and they're basically joined together. So the 2 polypeptide chains within each dimer, so the subunits, are held tightly together by strong hydrophobic interactions. And then there are ionic and hydrogen bonds between the alpha and beta dimers. So there's ionic and hydrogen bonds between these 2 here. Now, hemoglobin has various saturated forms depending on what it's saturated with or whether it's even saturated in the first place. So you have oxyhemoglobin here, and that's hemoglobin which is saturated with oxygen. We have deoxyhemoglobin, which is hemoglobin without a bound oxygen. We have carbaminohemoglobin, hemoglobin with carbon dioxide bound, and carboxyhemoglobin, which is hemoglobin with carbon monoxide bound. Now remember, carbon monoxide, the hemoglobin, the iron inside, has a higher affinity to bind carbon monoxide as opposed to oxygen as well. So that's just something to note. And finally, to finish off the video, there's various states of hemoglobin. They're known as T and R states. The T state means taut, and it's the deoxy form of hemoglobin. The 2 alpha beta dimers interact by ionic and hydrogen bonds, and what this does, it prevents movement of the polypeptide chains. So in this state, there is a low oxygen affinity, because there's little movement between the alpha and beta dimers. And the R state is the relaxed form, and this is where there is a rupture of some of the ionic and hydrogen bonds between the alpha and beta dimers, and this is due to the binding of oxygen. So what this means is that the polypeptide chains have more freedom of movement, and then obviously because of this there is a high oxygen affinity. That's everything just on the introduction of hemoglobin. In the next video, we'll be discussing various factors which affect the oxygen affinity of hemoglobin.